Hey everyone, it's your teacher here, Mr. Jennings, and we're at it yet again to talk to you about Mendel and some application of Mendel's concepts and a little bit more extensions to it. Just with these home videos, one thing to keep in mind is I'm doing these at home and I have two 4-year-olds, so they may make some noise while they're in the other room and they may bust in. So if you see awkward pauses or anything, or hear some playing in the background, you know what's going on. But alright, let's talk about Gregor Mendel and some of his extensions here. So when it comes to Mendel and genetics and probability, so we talked about this in class a little bit, but we want to make sure we got this. So probability is the likelihood that an event will occur. Scientists use probability to predict the possible outcomes of genetic crosses. For instance, if a coin is flipped once, the chances that it will land on heads is 1/2. And we can do this one here, even though it's not Nearpod. If it is flipped 3 times in a row, what's the probability of flipping all heads? Well, the idea is you multiply because it's an and situation. So when I go, what's the chance of me getting heads, heads, and heads? It's heads and heads and heads. So 1/2 times 1/2 times 1/2, meaning 1/8. So Punnett squares are the primary tool for doing this. When it came to the EOC, that Punnett squares weren't originally a straight standard. However, practice with it is vital. There will be a assignment with those. We kind of went over it in class, and I will post a video later, probably not today, showing you how to make Punnett squares. So that will be like the big extension activity for this week. So Punnett squares are used to represent all possible gene combinations that result from a genetic cross. Parent alleles are shown up on the top and the sides. These are parents in the blue boxes. And they separate because there is segregation of alleles. So you get one copy from each parent. And each box represents a possible outcome. When it comes to Punnett squares, you need to be familiar with some terms. Homozygous, meaning 2 identical alleles, either being capital T's or all lowercase t's. Heterozygous means 2 different alleles. We're going to have some static here. And then there's phenotype, which is the physical appearance, but it can also be an expression of a protein. It doesn't always have to be explicitly physical. It can be there in other ways. For example, though, tall and short, or phenotypes. All tall plants have the same physical characteristics, but when it comes to genotypes, this is the genetic makeup. And it could be big T, little t, big T, little t, or little t, little t. All tall plants may not have the exact same genotype because if you have any of the dominant allele, which is the capital T, you will express the dominant allele's phenotype. That means genotype can have some carriers. So things like homozygous and heterozygous more often than not will be appearing the same. That's why you cannot, when you look at a population, assume the genotype for a dominant individual because they could be homozygous or heterozygous. And that applies more to the AP level stuff when we talk about, when talking about stuff like Hardy-Weinberg. Alright, so a test cross. Now, Mendel used this to test the organisms of an unknown genotype. Basically, he would cross a plant with dominant but unknown genotype. So he knew it was either big T, little t, or big T, with a recessive plant. If the recessive plant appeared at all, he knew the plant was actually a dominant. Sorry, a heterozygous dominant individual instead of just homozygous, because if it was homozygous, it would have no short plants at all. So for a monohybrid cross, 1/4 of all F2 plants are going to be homozygous dominant, 2/4 will be heterozygous, and 1/4 will be homozygous recessive. The ratios tall to short will be 3:1, and this is the ratio Mendel found, and it's still used today. This is the traditional monohybrid cross. You take 2 individual hybrids that are monohybrid, mono meaning one, hybrid meaning mix. This is for one trait, in this case tall, and you combine the two hybrids for the tall. You combine 2 individuals that were hybrids for one trait, which in this case was tall. Now, probabilities can be used to predict averages. Now, probability can be used to predict the outcome of a large number of events, but cannot predict exact outcomes of a single event. So, for instance, I could say the likelihood I want to have, for example here, For one person, there's always greater outcome that they'll have a dominant trait, but this is not always true. In order to get results that reflect Mendelian ratio, a greater number of individuals should be considered. It's basically like flipping a coin. I can flip one coin and get heads 4 times in a row. That's possible. But if I flipped 1,000 coins, the chance of me getting 400 heads is very little. Just kind of keep it in perspective. The bigger the number, the averages are better. The probability becomes better. So probabilities really only work when you look at large picture, large scale. When you look at the small scale, you may see it deviate from the average, the probabilities pretty dramatically. Now, does the segregation of one pair of alleles affect the segregation of another pair? So this is what we call a dihybrid cross. I'm not going to show you how to make one of these, mainly because they're really not necessary to talk about for our standards. However, we do want to talk about them in terms of concepts. Basically, Mendel took a plant that was round and yellow in seed color and mixed it with, and it doesn't show here, round and green, round and yellow, another round and yellow individual, and found that the alleles for seed shape and color segregated, meaning individuals that were round could be green, and individuals that were wrinkled could be yellow, and they could also be green. And there was no linking there. Basically, round and yellow were independent of one another. And this is what we call independent assortment. So everybody has alleles that are segregated. That's through meiosis. But when you look at multiple alleles, more often than not, we typically find that a lot of traits are going to be independently assorted. Basically, you inherit them independent of each other. Inheriting one characteristic is not influencing the inheritance of another characteristic. And this is called a 9-3-3-1 phenotypic ratio. You can count the boxes. There's 9 boxes that are round and yellow. There's going to be 3 boxes that are wrinkled in yellow. There's going to be 3 boxes that are round and green. And there's only going to be 1 box that is wrinkled and green. And I'm going to move my face here. And you can see that. You can actually do the counting there if you wish. And how you make these boxes essentially is you just combine all the possible combination of gametes. So for instance, this one has, big R, little y, little y. You just figure out all the combinations of big R and little r, big R's and little y's and all that. And you get this. So for instance, this individual here, big R, big Y. That's one combination. Another combination is big R, little y. Okay, it's all the big R's taken care of. This one, little r, can go with big Y, and this one, little r, goes with little y. And that's all the combinations of little r. And that's how you fill these out. And this is what we call a 1, 2, 2, 1, 4, 1, 2, 2, 1, say 2, 1 genotypic ratio. We don't really do genotypic ratios when it comes to these. So independent assortment is the law here. This law states that genes for different traits can segregate independently during the formation of gametes. Independent assortment accounts for the genetic variety. So to summarize Mendel here, genes determine the inheritance of biological characteristics. In cases where 2 or more alleles of the same gene exist, some alleles are dominant, some will be recessive. Each adult has 2 copies of the gene, 1 from each parent. These genes are segregated, though, when gametes are formed. And alleles for different genes usually segregate independently of one another, meaning they independently assort. Goes to the end here. Alright, so let's kind of wrap things up. So when it comes to Gregor Mendel and his work, he basically, when he studied pea plants, the Punnett square didn't exist. Just think about that right away. It didn't exist. What he did discover, though, was over all these big mixes, he noticed that the ratios came out to be pretty much, in the case of one trait, he always got a 3-to-1 ratio. For every 3 dominant traits he saw 1 recessive trait, roughly. And the averages worked out weird. When he looked at 2 traits, like the round and yellow, he discovered that for every 9 round and yellow peas, which were dominant, he saw 3 that were round and green, and he saw 3 that were yellow and wrinkled, but also saw 1 that was both green and wrinkled, all recessive. So that's why we got the 9-3-3-1. And this is all about probability, the likelihood of events of happening. When it comes to the laws, the big thing to remember with Mendel is he discovered that one, this is coming back from the last video, that traits are inherited from your parents. They're called genes. 2, he didn't determine to call them genes, but he said traits are inherited from your parents. We now call them genes. 2, he discovered that some traits are are dominant to others. It's called law of dominance. 3, law of segregation, that you inherited one allele from each parent, and those alleles were segregated from each other during what we now know as meiosis. He didn't know that at the time, but we do now. And 4, the fourth law is the law of independent assortment. That the idea that usually genes are inherited independent of one another. So, for instance, the genes for round seed shape and seed color were independent of one another. Next time, we're going to talk about how to make a Punnett square just a little bit, just to have some extra practice. But then we will also talk about what happens when Mendel isn't right. Pretty much that there's going to be a lot of traits that we see that deviate from Mendel. And what does that mean? Alright, that's it. Thank you for watching. There will be some follow-up stuff on Canvas I recommend you work on if you're in my class. If you're watching this outside of my class, cool. See you. Take care. I'm Mr. Jennings. Have a great day.